Dear friends, in this video I'll talk about types of pollination mechanisms promoting cross-pollination. I'll begin with a picture of hermaphrodite flower. This consists of sepals, petals, androecium and gynoecium. When all parts of the flower are in the same flower, that is called as complete flower or perfect flower. The stamens contain anthers and filaments, and carpel consists, that is a female part, or gynoecium contains stigma, style and ovary. Coming to the petals, the function of petal, it is partly used to protect the male and female parts of a plant. Plants which use insects to transfer the pollen usually have larger petals which smell and are brightly colored to attract the pollinators. The male part, function of male part, it is called as stamen. It has long stalk called filaments. At the top of the filament is the anther which produces pollen. The function of female part, carpels are the female parts. They are made up of stigma, style and ovary. Stigma may be feathery or sticky to catch pollen grains. Ovaries, style is just to transfer the pollen from stigmatic surface to ovaries. Ovaries are where the eggs and seeds are produced. These are finally turned to fruits. How a flower is pollinated. The purpose of all flower is to be pollinated and produce seeds. Coming to the types of pollination, three types of pollination: self, often, cross-pollination and cross-pollination. The self-pollination, in this both anthers and stigma is present in single flower. Transfer of pollen from an anther to stigma of the same flower. Example: wheat, rice, barley, soybean, gram, etc. Often cross-pollinated plants. When cross-pollination takes place from 5 to 30%, then the crop is said to be often cross-pollinated. Example: sorghum, cotton, triticale, pigeon pea, tobacco. The third kind is cross-pollination. In this, transfer of pollen from an anther to a stigma of the different flower. Example: maize, sunflower, cabbage, tea, onion, carrot, papaya. Pollination is nothing but transfer of pollen from male anthers to female stigma. The mechanisms promoting cross-pollination are Uni Sexuality, dichogamy, hercogamy, Hetero Stylee, combinations of any of these above, then Self-Incompatibility and male sterility. For promoting self-pollination, cleistogamy, chasmogamy and all those things will help. But in case of cross-pollination, there are 7 mechanisms. In Uni Sexuality, there are 2 types, monoicy and dioicy. In dichogamy, protogyny and protandry. Coming to one by one, first one is the unisexuality, when only one sex organ develop in single flower. There are 2 types, monoicy and dioicy. Monoicy is a staminate and pistillate flowers occur in the same plant, either in same inflorescence like in mango, banana, coconut, or in separate inflorescence like maize, cucurbit and strawberry. In case of castor, that is also a monoecious condition, but the male and female flowers are positioned at different place. That is, the male flowers are placed above the inflorescence and female below that. Males are below and female is above. Dioicy, the male and female flowers are present on different plants. Example, in such species, male and female which are governed by a single gene. Example, papaya, hemp, date palm, etc. The maize plant representing monoecious condition, where the tassel or the male part and silk are at a different position. And another dioecious condition example is papaya, where we can find different plants, male plants and different female plants. This is a representation of dioecious plant. Example is holly. Dichogamy refers to maturation of anthers and stigma of the same flower at different times. It promotes cross-pollination even in the hermaphrodite species. Dichogamy is of two types. One is protandry and protogyny. Protandry is androecium matures earlier than gynoecium. Example marigold, maize, cotton, etc. Protogyny, gynoecium matures earlier than androecium. Example bajra. Coming to hercogamy, presence of a physical barrier or mechanical obstacles between anther and stigma ensures cross-pollination. Example calotropis gigantea. Hetero Stylee. When style and filaments in a flower are on different lengths, it is Hetero Stylee. It promotes cross-pollination. Example Primula vulgaris. Two types of flowers in Primula, pin and thumb. Pin flowers have long style and short stamen. Thumb flowers have short style and long flowers. This characteristic is governed by a single locus S. Allele capital S is dominant over small s. But the characteristic of Hetero Stylee or pin and thumb kind of flowering is governed by recessive locus S. This is a Hetero Stylee. In the first picture, there are small anthers and long stigma or style, and the gynoecium portion is long. But here the anthers are short, long, and stigma is short. This is a typical pin and thumb flower in Primula. Can see here the gynoecium part is long in the first picture, whereas in the second one anthers are longer than gynoecium and gynoecium is short here. Coming to self-incompatibility. It is the failure of pollen to fertilize the same flower or other flower of the same plant. It is failure of pollen tube to penetrate the full length of style and effect fertilization. Self-incompatibility is an important outbreeding mechanism which prevents autogamy and produces allogamy. It has reported in more than 300 species belonging to 70 families. In self-incompatibility, non-fruitfulness or failure of fruit setting may occur due to pollen grain fails to germinate on the stigma. Pollen grain germinates, but pollen tube fails to enter inside the stigma and style. Sometimes pollen tube enter inside the style, but growth is very slow. Pollen tube enters the ovule, but there is no fertilization due to degeneracy of egg cells. Sometimes fertilization is affected by embryo degenerate at very early stage. The classification of self-incompatibility was given by Lewis in 1954. According to him, there are two types, heterotrophic and heteromorphic and homomorphic. Heteromorphic are two types, distyle and tristyle. Homomorphic means gametophytic self-incompatibility and sporophytic self-incompatibility. Gametophytic is again divided into monofactorial and difactorial self-incompatibility. The major difference between gametophytic and sporophytic, that is homotypic self-incompatibility. In gametophytic, the outcome of the interaction between pollen tube and style is determined by genotype of the pollen, that is gamete. Here S locus products are synthesized after completion of meiosis. The growth of pollen tube arrests in the style. In case of sporophytic incompatibility, the outcome of interaction between pollen tube and style is determined by genotype of the sporophyte, that is a diploid tissue. Here S locus products are synthesized before completion of meiosis. The growth of pollen tube arrests at the surface of the stigma. The sporophytic is may be compatible or incompatible, but in gametophytic system the things are compatible, incompatible and partially compatible. In this picture you can see in gametophytic system the S1 and S2, again the pollen type is S1 and S2, and the stigmatic surfaces or the genes governing gynosium are also S1 and S2, that leads to incompatible reaction, whereas S1, S2 and S1, S3 interaction is partially compatible, and the another interaction S1, S2 into S3, S4 are fully compatible. In case of sporophytic system, here the incompatibility is governed by multiple alleles, so the dominant gene governs the other dominant loci governs the other one, like S1 governs the S2. Likewise, when S1 S2 into S1 S2 interaction happens, that lead to incompatibility, and only S1 S2 into S3 S4 gives compatible reaction. Coming to male sterility system, it is defined as absence or non-functional pollen grains in plants or incompatibility of plants to produce or release functional pollen grains. The use of male sterility in hybrid seed production has a greater importance as it eliminates the process of mechanical emasculation. Classification of male sterility. There are four types: genetic, cytoplasmic genetic and chemically induced. The another is pistillate kind of male sterility found in castor. In genetic male sterility, temperature sensitive, photoperiod sensitive, transgenic male sterility. The another type is cytoplasmic genetic and chemically induced. Comparisons among the three: in GMS, it is controlled by nuclear gene, and CMS cytoplasmic and CGMS by nuclear as well as cytoplasmic genes. GMS consists of AB lines, CMS also AB lines, and CGMS A, B and R lines. GMS is affected by temperature, whereas CMS and CGMS are not affected. CGMS is used in production of hybrids, both seeds and vegetatively propagated. CMS is used only for vegetatively propagated, and CGMS for both vegetatively and seed propagated plants. The interaction in GMS is recessive MS-MS into heterozygous MS-MS form. In CMS it is A into B, and in CGMS it is A into B. In GMS, the male sterile and fertile ratio is 1:1. CMS is vegetative type, and in CGMS the sterility is ABR type, where A and B lines are crossed in order to maintain the male sterility or maintaining the female lines, and R is used as a pollen source, and when it is crossed or A line is crossed with B, that will give hybrid seed. Example of GMS used in barley, wheat, tomato. CMS onion, sugarcane, forage, and CGMS in pearl millet, sorghum and cotton.